Hi everyone, it's Dr. LeBlanc. This video capture lecture is on DNA replication, Meselson and Stahl's experiment. Our learning goals are to understand the structural elements of the DNA molecule that led to Meselson and Stahl's results, that DNA replicated semi-conservatively, and to understand the design and execution of their experiment. This is the model of DNA replication showing a replication fork, the two parental strands here in blue and yellow, and the newly synthesized DNA strands in purple. DNA replication relies on the complementarity of the DNA strands. The ATGC rule, or Chargaff's rule, where A base pairs with T with 2 hydrogen bonds and G base pairs with C with 3 hydrogen bonds. The two DNA strands come apart. Each serves as a template for the synthesis of new strands. The two newly made strands are called the daughter strands, and the two original strands are called the parental strands. Here is a diagram showing just that, with the two parental strands in purple and the two new daughter strands in blue. Here is our replication fork. When the two nucleotides come apart at the replication fork, they are open to be hydrogen bonded by new incoming nucleotides. They come in one at a time and add on to the growing 3' end of the DNA molecule. Remember that the two strands of DNA are anti-parallel to each other, so this end of the new strand is the 5' end here, and here's the 3' end. So if this is the 5' end of our new strand, all the way up here is our 3' strand. DNA replication always proceeds in a 5' to 3' direction, so new nucleotides get added to the growing 3' end of the molecule. We can distinguish two different DNA strands that are newly synthesized. The leading strand, which is synthesized continuously as nucleotides are added on to the growing 3' end, and a lagging strand, which is synthesized in discrete segments of DNA that are then ligated together. So here is our parental strand here. Here are two new molecules of DNA. They are identical in sequence to the original DNA strand. There were 3 proposed models of DNA replication in the 1940s. There was a conservative model which proposed that the parental DNA strand stayed intact and produced a new molecule of double-stranded DNA where all of these nucleotides were newly synthesized. The second model, the semi-conservative model, proposed that the two parental strands came apart and each of the parental strands acted as template. That's the model that we just discussed in the previous slide. This is the model that won out. Finally, there was also a dispersive model which stated that the parental strand somehow broke up into little pieces and was incorporated into the newly synthesized DNA strands, so that a new DNA molecule had two strands that were each a mix of both parental DNA and newly synthesized DNA here in the light blue. There are hypotheses that Frank Stahl, Matthew Meselson, and working in Arthur Kornberg's lab came up with was to design an experiment to test those 3 models. This is what they're doing now, and if you want to learn more about each of these individuals and hear an interview, especially with Matt Meselson, about how they made a mistake in labeling their data, you can go to the DNA from the beginning.org, which is at the Cold Spring Harbor DNA Interactive website. So this is their experimental design. The key to their experiment was that they used two different isotopes of nitrogen. Normal, every run-of-the-mill nitrogen is N14, okay? But you can also make a heavy isotope of nitrogen, N15. If you grow your E. coli in a medium containing heavy nitrogen for many generations, the DNA that is in those cells will be heavy, will have the heavy isotope of nitrogen incorporated into their DNA. That was the starting point for the Meselson and Stahl's experiment. They took some E. coli cells that had been labeled with N15 and added an excess of N14 solution, and then let the bacteria replicate normally. At each generation time, they removed some of the cells from this culture and extracted the DNA and put it on a gradient of cesium chloride. Cesium chloride is a very heavy salt. If you make a solution of cesium chloride and put it in a test tube and spin it very hard and very long, 100,000 times G for 3 days, it will create a gradient of density inside the test tube where the bottom of the test tube is very heavy and the top of the test tube has a lighter density. The DNA mixed in this solution will all settle in the test tube at the same density as the salts around it. So if you have a heavy DNA molecule containing N15, you will find it more towards the bottom of the test tube. And if you have a light molecule of DNA with N14, it will remain closer to the top of the gradient, where the lighter density is. Okay, so that's what they did. They mixed their DNA preparation with the cesium chloride gradient, spun it really hard for a very long time, and then carefully removed the test tubes from the centrifuge and shone ultraviolet light on them, which will detect the DNA in the test tube. The DNA within the gradient can be observed under ultraviolet light. The light DNA will be towards the top of the test tube, and the heavier DNA will be towards the bottom of the test tube. What they observed, Meselson and Stahl observed, was this pattern of light DNA and half-heavy DNA after 2 rounds of DNA replication. This is the result shown here is after 2 generations. Let's review what the model said. Now, if the purple strands are the heavy N15 containing DNA strands, the conservative model would state that after the first round of DNA replication, you would see a heavy molecule and a light molecule. And the semi-conservative and the dispersive models would suggest that you would see not a heavy molecule and not a light molecule, but an intermediate, a half-heavy, half-light molecule. These DNA molecules are also half-heavy and half-light. They have a different configuration, but their density is still half-heavy, half-light, half-heavy, half-light. So after the first round of DNA replication, you could rule out the conservative model, but you couldn't distinguish between the semi-conservative and the dispersive. To do that, you would need to go to a second round of DNA replication. Then you would see a distinction between the 2 models. The results predict that if you see a half-heavy band and a light band, that is consistent with the semi-conservative. However, if it was dispersive model, you would only see one band of DNA, which was lighter than this band of DNA. This is the results they got. After 1 generation, the DNA is all half-heavy. Here it is, right here. Here is your band of DNA. Initially, you start out with some heavy DNA before it's replicated. That's the parental strand. But as the cells grow and DNA is replicated, you begin to see this half-heavy, and then after a single generation, you only see the half-heavy DNA. This is consistent with both the semi-conservative and the dispersive models. However, after 2 generations, the DNA is of 2 types, light and half-heavy. That is only consistent with the semi-conservative model. And so Meselson and Stahl were able to rule out both the conservative model and the dispersive model and showed that the mechanism of DNA replication is consistent with the semi-conservative model of DNA replication. This is their actual notebook data from their notebook. 1 generation, 2 generations. This is a density reading from this film. So in summary, we've learned that DNA replicates semi-conservatively. We learned that the 2 strands of a parent DNA molecule separate by breaking the hydrogen bonds between the complementary base pairs, and that each parent strand acts as a template and remains intact. Meselson and Stahl's data is consistent with a model that the newly synthesized strand is synthesized from complementary nucleotides that hydrogen bond to the template strand.